The CLEOPATRA study also is going to be presented, but the results are already out. They came out in the New England Journal of Medicine today as a special edition. And this is a study that is taking patients with HER2-positive advanced breast cancer and comparing what would be a standard regimen of Taxotere plus Herceptin and adding the antibody pertuzumab. So pertuzumab is another antibody to HER2. It binds at a slightly different part of the receptor, and it inhibits the receptor from functioning in a proper way. And this is a conceptual advance in that it tells us that we might get a benefit from blocking two parts of the same pathway. Because we know that with Herceptin alone, just about everybody eventually develops resistance in the case of advanced breast cancer. Pertuzumab was shown to improve the time to progression, and so this is definitely an important achievement because it tells us that we have the potential to keep going. So we know that cancer cells find ways to become resistant when you block a growth pathway like you do with Herceptin. We don't fully understand the mechanisms, but we do know that if you block different parts of the pathway, since it's sort of like a charge relay, the signal goes through many proteins, and if you block several of them, you may actually get a much bigger effect. So just as is in the case with hormonal therapy, with the HER2 system, one can do that as well. So the fact that pertuzumab and trastuzumab work well together and improve disease-free survival and maybe even overall survival, that data is not out yet. There's a hint of fewer deaths, but it's too early for that analysis. But if that ends up being true, we can build on this. We can identify other mechanisms of resistance and start using the right combinations to really make even bigger impacts.